Very briefly in English, I would like to welcome you all to this third edition of our webinar series on understanding the human microbiome. It's really great to have you here with us today and that you are so interested about the microbiome. We have organized translation from English to German and vice versa for this session, so I will switch to German in a minute. But first, for those of you who would like to follow this session in English, you can listen to the translation by choosing the English interpretation channel in Zoom. Just check at the bottom of the application. You should see a symbol there for the interpretation. Click on it and choose your preferred language. So, und jetzt noch einmal auf Deutsch. Herzlich willkommen an alle Zuhörerinnen und Zuhörer, an alle Interessierte zu unserem dritten Webinar der Reihe Understanding the Human Microbiome, also das menschliche Mikrobiom verstehen lernen. Diese Webinarreihe veranstalte ich gemeinsam mit der UEG, mit der United European Gastroenterology. Und ich finde, das ist eine großartige und wichtige Sache. Zuallererst einmal geht wieder mal ein großes, herzliches Dankeschön an die Unterstützung der UEG, dass wir diese Webinarreihe überhaupt durchführen können. Vielen Dank. In Vertretung hier Dr. Markus Beck, mein Co-Moderator, der nach mir gleich reden wird. Wenn wir vom Mikrobiom sprechen, meinen wir damit die Gesamtheit in unserem, aller Mikroorganismen in unserem Darm. Diese Organismen oder Bakterien leben in einer symbiotischen Beziehung mit uns und helfen bei der Verdauung, nicht nur, aber auch, solange alles in einer gesunden Balance miteinander steht. Wenn diese Balance gestört wird, kann das auch ganz schnell zu gesundheitlichen Beeinträchtigungen oder dann eben auch zu folgenschweren Krankheiten führen. Lange wurde dieses Mikrobiom aber gar nicht allzu große Aufmerksamkeit geschenkt und erst in den letzten, kann man sagen, 10, 20 Jahren haben wir ernsthaft mit der Erforschung des Mikrobioms begonnen und für die Bedeutung für unsere Gesundheit. So wissen wir heute, dass das Mikrobiom einen viel größeren Einfluss auf unsere Gesundheit, auch auf die seelische zum Teil und die geistige hat, als wir bisher angenommen haben. Das ist auch nicht verwunderlich, denn wir sind keine Individuen, sondern wir sind viele. Wir haben mehr Mikroorganismen als eigene Zellen. Studien zeigen, dass das Mikrobiom auch bei der Tumorentwicklung eine Rolle spielt. Deshalb werden wir uns heute mit folgenden Themen beschäftigen. Erstens, das Potenzial des Darmmikrobioms bei der Verhinderung der Tumorentwicklung. Zweitens, die Rolle des Darmmikrobioms bei der Entstehung und dem Fortschreiten bösartiger Verdauungserkrankungen. Und drittens, die Rolle des Darmmikrobioms bei der Behandlung von Krebs. Sie können die ersten Webinare aus unserer Reihe auf der Homepage von UEG nachhören oder auf meinem YouTube-Kanal Sarah Wiener. Dabei ging es zum Beispiel über Lebererkrankungen und Fettleibigkeit oder das Mikrobiom und schwerst verarbeitete Nahrungsmittel, was besonders bei mir als Köchin natürlich ein besonders spannendes Thema ist. Wir haben heute wieder zwei hochrangige Experten eingeladen, um dieses Thema heute zu diskutieren. Zum einen Professor Thomas Seufferlein, Präsident der Deutschen Krebsgesellschaft. Er wird uns einen Überblick über die Verbindung zwischen Darm und Krebs aus der Sicht der Onkologie geben. Und Professor Herbert Tilg von der Medizinischen Uni in Innsbruck wird spezifisch über den Zusammenhang zwischen dem Mikrobiom und Darmkrebs reden. Bevor wir von den beiden Experten hören, möchte ich das Wort aber noch an meinen Co-Moderator abgeben, Dr. Markus Beck, Vorsitzender des Ausschusses für öffentliche Angelegenheiten des UEG. Lieber Markus, the floor is yours.
Sarah, thank you very much for the nice introduction. Again, UEG is very thankful to you that you're hosting this event. Because everybody knows without you this would be impossible to do. And as you said, it's the third time we're doing this. We had really outstanding speakers and presentations so far, and today will be no exception to this rule. Before we start, I will give you a brief introduction into what UEG is here. UEG, United European Gastroenterology, is a scientific umbrella organization aiming to improve digestive health. And UEG is uniting 30,000 specialists from every field of digestive health to form a very powerful and knowledgeable organization. Now, the microbiome, of course, is interesting for almost every medical discipline these days, and it's one of the major focus topics in research. Obviously, the gut microbiome is very important for digestive health. And when we look at the groups within UEG that are working on that, we actually have two groups that are very actively involved in research here. The European Helicobacter and Microbiota study group, a multidisciplinary group founded by pioneers in the field, originally just working with one bacteria, with Helicobacter, but now in recent years having expanded to the whole microbiome. They do have an official journal, Microbiota and Health and Disease, and they do have an annual conference on microbiota therapeutics, writing guidelines here, and are also working on cancer early detection and prevention. And then, of course, very importantly is the European Society of Neurogastroenterology and Motility, which is focusing on neural, endocrine, metabolic, immunological, and microbiological regulation of gastrointestinal functions in health and disease. Originally, this was called the functional GI disorders, and 1 in 10 Europeans is actually affected by problems here, oftentimes at young age. But as we understand more and more about the gut and the microbiome, we understand that many of these diseases that formerly were called functional are actually have something to do with the metabolic function also of the microbiome. And therefore, projects which they're running, like the Gut Microbiota for Health project, is about knowledge sharing and debate promoting gut microbiota for these very common disorders. And then we do have other specific microbiome-focused digestive health research projects, like this legacy project funded by the European Union, which is a European consortium for a personalized medicine approach to gastric cancer. And here you can see the different work packages that are involved here. It aims to improve gastric cancer outcome and to apply personalized medicine to its solution. So with that, the only thing that I would also like to highlight here, which does not necessarily have something to do with the microbiome, specifically, is the Awareness Week group, which is currently ongoing in Brussels, and I would like you to take note of that, because I think this is a very important topic that also has something to do with the gut microbiome. Now, as Sarah has already told you, we had fantastic speakers in the last two webinars, and we have terrific speakers again in this webinar series. And the first one will be Professor Thomas Seufferlein from the University of Ulm to give his talk on microbiota and cancer. So with that, I'd like to hand over to Thomas. Thomas, please.
Yeah, dear Markus, thanks very much indeed. And dear Sarah Wiener, thanks very much for the opportunity to present this overview on cancer and the gut microbiota. And my really task is to give you a general overview. And later on, my dear friend and colleague, Herbert Tilg, is going to give you specific insights on colorectal cancer. The next, please. I don't have any. Next, please. Can you give me the next slide, please? Okay, I don't have any conflicts of interest for this presentation. Next one, please. And when we look at our microbiome, we have about 10 to 100 trillion microorganisms in the human body. And they encode more than 3 million genes and produce thousands of metabolites that modulate the human host. And they're adapted, as you see on the right side, very nicely to the intraluminal pH, for example. We have a very acidic pH in the stomach, and it's getting more and more neutral as we go on in the small bowel and the large bowel. There are about 500 to 1,000 unique species per individual. And what do? These microorganisms? They produce and break down food molecules into nutrients. So they are quite important for digestion and produce fatty acids, vitamins, and also amino acids. And they're also important for the integrity of the immune system and the gut lining. So the integrity of our gut lining is quite dependent on a good functioning microbiome. And diversity of the microbiota is really key in this context. Next, please. Next one, please. So when we look at the evolution of the gut microbiome, we see that the microbiome is seeded in utero. And it takes a while, up to 3 to 5 years of life, until we actually adopt a more or less adult-like microbiome in our body. The normal microbiome is constitutes of two major phyla, the Bacteroides and the Firmicutes. The older we get, the more likely we are to actually get so-called dysbiosis. So old age comes along with many other things, but also with a change in the microbiome. And this might be a link also to why cancer originates predominantly at an advanced age. The next slide, please. Next, please. And our microbiome knows where we live and how we live. And this is quite interesting. This is one example. When you migrate from a country like South America to the US, this actually can lead to a lower diversity of the gut microbiota. This reflects changes in the environment, but also changes in lifestyle. And this might be an explanation why the incidence of particular diseases, including obesity and some cancers, increases in populations of US immigrants compared to their country of origin. And studying this phenomenon could provide new clues in the role of microbiomes in actually promoting cancer. The next slide, please. Next one, please. Yes. And one important thing is our microbiome is really a self-portrait of ourselves. So for example, exercise is linked to an increase in the number of beneficial, good microbial species and enriched microbial diversity. So we have better short-chain fatty acid synthesis. We have better carbohydrate metabolism, and it works already by increasing the frequency of moderate endurance exercise, in this case from never to daily. 150 minutes per week, that's not too much. So if you take the stairs instead of taking the elevator, you might already come up to the 150 minutes per week. So that's worthwhile. The body fat percentage, the muscle mass, the physical activity are significantly correlated with certain bacterial populations. And we know that Akkermansia, for example, do play a role in that. So our microbiome is not static, it's continuously changing. The next slide, please. The next one, please. Yes, and a lot can go wrong at the level of the microbiome during life. And this is interesting. A lack of exercise, stress, antibiotics change the microbiome. We already talked about older age, decreased gastric motility. Our diet changes substantially the microbiome. Even the mode of delivery. So when we have a normal vaginal delivery, the microbiome is totally different from when we have a cesarean section. And this can affect even our microbiome in later life, which is quite interesting. And of course, also genetic effects of the host, of ourselves, do play a role. The next slide, please. So microbial alterations, the so-called dysbiosis, are implicated in many diseases. And you heard from Sarah Wiener already that obesity, fatty liver disease, diabetes, inflammatory bowel disease, but also cancer are related to that. And alterations to the microbiome are associated with cancer development, with cancer progression, but also with drug response. And I will give you some examples. The first links, interestingly, between cancer and microbes dates back already 4,000 years. So this is not something we invented in the last 50 years. This is really, there will be quite clever people 4,000 years ago that drawn the link. Interestingly, this ranges from the diagnosis to prognosis to treatment as well as remission. Blood-borne microbial DNA, for example, can be used to diagnose certain cancers. The next one, please. Next slide, please. The next. Perfect. So there are a few microbes that directly cause cancer, and only 11 microbial species are really oncomicrobes, human carcinogens. But interestingly, they cause an estimated 2.2 million cancer cases per year, 13% of the global cancer cases. For example, Helicobacter pylori is one of these oncomicrobes, but the majority of these bugs complicit in tumor growth. So they act through the host immune system and modulate primary and secondary lymphoid tissues. They also produce metabolites that act on cancer, and they cause antigenetic mimicry with cancer cells. Not only bacteria itself, but also fungi and bacteriophages contribute to gastrointestinal cancers. The next slide, please. So the potential mechanisms of dysbiosis, supported tumorigenesis, and the tumor growth are, for example, bacterial-produced secondary bile acids and chronic inflammation. In the stomach, it's Helicobacter pylori. As you see below, this is the bug, and of course, you know the Nobel Prize has been awarded to Beatty and Warren for discovering that Helicobacter pylori causes gastric ulcers, but also gastric cancer. The next one, please, that you see, this is a breakdown of the mucosal barrier. Can you just move on one slide? Exactly. So we have this no, back one, please. This was an animated slide. Sorry. Exactly, that we are here. You have a microbiota in the mucous layer on top of the intestinal epitheliums, and a breakdown of the mucosal barrier causes damage there, and also exposure of the epithelium to toxins with genotoxic properties. Bacteria can induce oncogenic transcriptional activity and can impair anti-tumor immune functions. And in breast cancer, for example, the microbiota can even affect the levels of hormones and alter the balance in energy metabolism. The next slide, please. So, one example in the skin, the effect of the microbiome on cancer can be either directly or indirectly. In the direct effect, we have an interaction of the microbiomes, they're residing in tissues where the cancer emerges, such as the skin, interacting with melanoma. But interestingly, we also have bacteria in tumors. We have bacteria in colorectal cancer, as you will hear later on, in pancreatic cancer, in liver cancer. There is an intratumoral microbiome. And you have, of course, the direct effects, but you also have indirect effects, but interaction between microbiota and the cancer that resides in a different tissue. And these long-range effects, for example, are caused by metabolites that actually promote progression of skin cancer or its response to treatment. And diet, interesting, can play a role in this context because it affects the circulating levels of metabolites and the microbiota composition in the bowel. The next slide, please. Next one, please. And this is interesting because even metastases have the same bacteria as the primary tumor. And when you have tumor cells that actually metastasize from the primary tumor to a distant organ, they carry bacteria from the primary tumor to the distal tissues. So the same bacteria are found in, for example, colorectal cancer in the primary as well as in metastases, and they travel alongside with the metastatic cells. Interestingly, this is data from mice. Antibiotic treatment could reduce the tumor load in this context, and maybe that's some potential for future treatment of patients. The next slide, please. So when we think about, finally, how the microbiota change treatment, there are several ways of how microbiota affect the treatment. For example, 5-FU is affected by bacterial vitamin B6, B9, and ribonucleotide metabolism. And the microbiota can actually degrade 5-FU in such a way that it's not efficacious. On the other hand, cyclophosphamide efficacy is critically dependent on two bacteria, Barnesiella and Enterococcus hyrae. Without those, cyclophosphamide doesn't work. And even things like immunotherapy, which is now a real rush in cancer treatment nowadays, actually depends on Bacteroides species that actually colonize the mucosal layer and induce T helper cell immune responses, and thereby support and enable the treatment efficacy in the first place. So microbiota play a huge role in the efficacy of even chemotherapy, and they can affect it both ways, either supporting it or antagonizing it. The next one, please. So when we look at immunomodulation, then mucosal microbes can have a major impact on the immune system, either locally, they can translocate to the sites of growing tumors, or via the mediators like metabolites, cytokines, chemokines, toxins, and even bacterial released vesicles. And the microbes can directly with the immune cells or indirectly via immune modulation and inflammation. And again, the inflammation caused by the microbiota can be either pro-tumorigenic or can actually inhibit the tumor. And they have a very diverse range of effects of the innate and adaptive immune system. So as a matter of fact, microbes work on anything and everything in the human body. The next one, please. And it does even more. This is an interesting experiment. This is a mouse model where they actually could show that the probiotic Lactobacillus acidophilus strain can decrease pain relative to controls and can potentiate the effect of opioid analgesics. So your microbiota may even be able to modulate painkillers in patients, and particularly that's important also for cancer pain. So you see there is a huge range of effects microbiota and bacteria in the bowel can have. The next one, please. So when we look at therapeutic strategies, can we use that to modulate the cancer treatment? It's not yet clearly established. We can target the microbiota selection. We can use probiotics, prebiotics. We can do fecal microbiota transplants, or could use antibiotics. That's actually quite used now in clinical trials. Of course, we can target the diet by fibers, by fermented foods, by plants, by polyphenols. Green tea has a lot of polyphenols, epigallocatechin gallate, for example, and that has quite good effects in preventing, for example, colorectal cancer. And we can, of course, otherwise modulate the microbiome, as I told you, by exercise. The next one, please. So in conclusion, our gut microbiome is a self-portrait of ourselves. It's highly dynamic in changes in lives and responds to many factors, including diet and exercise. It can become dysbiotic and thereby contribute to tumor development directly and indirectly via metabolites, also to metastases and to treatment resistance. On the other hand, it can be targeted and be used to improve treatment and ideally to prevent cancer. The next one, please. Next one, please. And as with many things in life, it also holds true for the gut microbiome. Diversity is key in this context. And with that, I thank you very much for your attention, and I'm happy to take questions later on. And hand over to Herbert.
Herbert, you need to unmute yourself. You're still on mute, Herbert, please.
Lieber Herbert, Sie sind gemutet.
Now it should work.
Yes, now you're here.
So sorry for this short delay. In the second lecture, you will hear now about the gut microbiota in colorectal cancer. I just try to move the slides forward. For whatever reason, it doesn't work. Now it works. So I will start my lecture with an interesting mouse model we have developed a few years ago. And you can see here that certain knockout mice, and we worked with lipocalin-2, which is an important immune mediator, that they have no gut inflammation. But as soon as you generate mice which have also a deficiency in IL-10, they develop inflammation. And we have many diseases in humans where inflammation plays a major role in the later development of colorectal cancer. So in those so-called double knockout mice, IL-10 plus lipocalin-2, we see a lot of inflammation and many pro-inflammatory cytokines are upregulated. But more importantly, those mice develop tumors. So it's not only an increase in tumor numbers, but also in tumor area. And this means we have a model which has the sequence of nothing, then they develop inflammation, and then they develop tumors. Why is this important? Because we have many diseases in humans, especially in the intestine. For example, inflammatory bowel diseases which affect in Europe 1% of the population, mainly in the third life decade. And one of the major complications, is inflammation-associated tumor development in the colon. Well, we know in this model that when we use antibiotics, we can improve inflammation, but we also know in this model when we improve inflammation with antibiotics, we can reduce tumor load. And we could identify two bacteria, mainly one bacterium, Alistipes finegoldi, and also Robinsoniella. This means there are certain bacteria which under certain circumstances are able not only to drive inflammation, but also to drive cancer. And this highlights the important role of the gut microbiome, potentially also for tumor development. What can we say about Crc and associated bacteria? As you can nicely see in this cartoon, and usually now we speak here about spontaneous colorectal cancer or non-inflammatory associated colorectal cancer. And you can see usually this type of cancer develops from the normal epithelium. We have a hyperproliferative epithelium, then we have an adenoma, and finally we have an adenocarcinoma. And as you can see here in the case in the development from adenoma towards adenocarcinoma, we have a lot of immune cells in the tumor tissue, but we have a lot of various microbes also within the tumor. And I will speak about three bacteria which at the moment are the most attractive and important bacteria playing a role in this disease. It's first E. coli, namely a very special E. coli, a colibactin producing E. coli. Then in the second case is enterotoxigenic Bacteroides fragilis. And in third case, it's Fusobacterium nucleatum. We have a lot of data from epidemiology, but we also know that all those microbes are enriched in this cancer, and also some mechanisms have been identified. This is one of the first reports published in BNS in 2014, where they demonstrated that in colorectal cancer, we see especially in the right colon a so-called biofilm. Now, a biofilm is a very complex biochemical process, but within the biofilm, we can mainly see a bacteria. So the biofilm is mostly generated by various bacteria, and they have shown, for example, here in right or in left-sided colorectal cancer, that this biofilm contains, for example, mainly bacteroides. So biofilm is a major aspect which has been demonstrated to be of relevance in colorectal cancer. But this has also been demonstrated for other diseases, for example, for a disease which is called FAP. FAP is a genetically driven disorder where almost 100% develop polyps. And in this science paper, they have shown that also in these FAP patients, they could observe the so-called bacterial biofilm. And in this report, they could demonstrate that within this biofilm, you can find mainly, again, the before mentioned E. coli, which is demonstrated in red, and Bacteroides fragilis, which is marked in green. So biofilm is obviously something which is of relevance in colorectal cancer. And this genetic disease, FAP, is a very excellent example into such a direction. And they have demonstrated, for example, when you use those bacteria, as demonstrated in the middle of this graph, which contain E. coli, especially E. coli, when you co-colonize mice with those bacteria, both E. coli plus Bacteroides fragilis, then this causes more tumors with respect to number and also affects mortality in such a murine model of colorectal cancer. We have performed one of the first studies together with Chinese colleagues assessing the gut microbiome along the colorectal adenoma carcinoma sequence. It was one of the first metagenomic studies, meaning we did a deep sequencing within the material we collected. And in this study, we had 55 healthy controls, 42 patients with advanced adenoma, so no cancer yet, and 41 patients with colorectal cancer. As you can see in the upper part of the slide, that the G number in cancer was much higher than that in controls, and the colorectal cancer patients are here outlined with red. And this is, of course, a very busy slide, but it tells us on the left side the healthy controls, and here the cancer patients. And as outlined on the right part of the slide, we had a couple of bacteria which were convincingly associated with the appearance of a colorectal cancer. And it's interesting because here you can see, for example, again, a bacterium with the name Alistipes putredinis. And as you remember in my first slides in our animal model, where we demonstrated the sequence inflammation colorectal cancer, we also demonstrated that a certain Alistipes is important within this disease. We advanced our clinical data with colleagues from China, with a large series of patients from Denmark. We used published cohorts, our one and from France, and an additional Chinese cohort, and did again a metagenome-wide association study, which was then published in Gut in 2017. And in this study, in this collaborative study, we could identify further interesting bacteria besides Fusobacterium nucleatum, namely two bacteria entitled Parvimonas micra and Solobacterium morei. And as you can see here, we were able to identify biomarkers which allowed us to stage the disease. For example, this was one biomarker, so-called butyryl coenzyme A dehydrogenase from Fusobacterium nucleatum, which correlated with stage of disease, or also with a genetic marker from Parvimonas micra. This means we have now a couple of interesting bacterial candidates, which might be involved in colorectal carcinogenesis, and we have observed robust or identified robust gene markers associated with colorectal cancer. And all our data went into a large Nature Medicine paper from 2019, where they used the available cohorts published at that time and could again convincingly shown that there is indeed a very clear microbiome signature in colorectal cancer. And for example, they could link it with an interesting pathway, namely choline degradation. So overabundance of choline trimethylamine lyase gene was associated, highly associated with colorectal cancer. We have already heard in the lecture before that we can find colorectal cancer tissue-specific bacteria not only in the primary lesion in the colon, but also in the colorectal, in the respective liver metastasis. And this has been shown in this science paper published 2018, and they could demonstrate that in all subjects who had Fusobacterium in the colon, in the primary lesion, They also found Fusobacterium nucleatum in the liver metastasis. Now this is interesting. Of course, we do not know exactly how those bacteria could move into the liver, and are they really relevant, or are they only bystanders and do not play a role in the generation of metastasis. But anyway, it is an interesting aspect and an interesting new aspect in carcinogenesis and respective metastasis. Now, the findings I mentioned are summarized here in a cartoon from a review paper we published in Cancer Cells. So I mentioned Fusobacterium nucleatum. You can see here a structure which is typical for the colon. And you can see also that Fusobacterium is activating certain pathways which play a role in colorectal carcinogenesis, for example, the beta-catenin signaling pathway. But also other bacteria, as I mentioned, are important, like this BKS positive E. coli. We also know that Bacteroides fragilis is involved, for example, via activating of reactive oxygen species production. And I mentioned other bacteria like Alistipes, which has been demonstrated not only in animal models as stated, but also in large clinical cohorts. They probably all also contribute to a state of chronic inflammation, which is involved in many states of colorectal carcinogenesis. Dear colleagues, dear ladies and gentlemen, let me summarize. There are models around which nicely have demonstrated the sequence of inflammation and cancer, and obviously bacteria are here involved like Alistipes. It's very clear now that colorectal cancer is associated with profound alterations in gut microbiome, and colorectal carcinoma is probably the best studied cancer at the moment with respect to potential roles for the gut microbiota. There are several bacterial strains which have now been identified, potentially supporting CRC development. I mentioned the names, and we have identified a new bacterium, Parvimonas micra, in the last years. The gut microbiome affects efficacy of systemic treatment with immune checkpoint inhibitors. That's another fascinating area in medicine at the moment, and we are currently in the stage of learning a lot of things in this part of medicine. And overall, bacteria, intratumoral bacteria, is definitely a hot area in cancer research at the moment. Thank you very much for listening.
Thank you very much, Herbert. Great presentation. And we would like to start the discussion. Sarah, would you have a question to start with?
Ja, vielen Dank an diese spannenden Vorträge. Ich habe erstmal eine ganz primitive Frage an, die sicher auch die Zuhörenden interessieren werden, die jetzt nicht so tief einsteigen können, wissenschaftlich, wie wir das jetzt bei Professor Tilg gehört haben, und die vielleicht als allererstes interessiert, was könnte dann jeder persönlich für sein gutes Mikrobiom machen und sozusagen vielleicht auch als Prävention gegen Krebs und Krebstumore? Was würden Sie empfehlen?
Ja, wenn ich anfangen soll, ich habe versucht, I tried to do it in English. I tried to explain that in my talk. I think exercise is quite important, and you don't have to do much. It's only 150 minutes per week that already has a substantial change on your microbiota. And of course, what you eat is what you are. So the right kind of food, fiber, also bearing in mind that polyphenols are good for your microbiota, and actually it helps you to select the right ones as compared to the wrong ones, which means, for example, containing green tea extract and stuff like that. So all these things you can do to really improve your microbiome and to really do something about it. So eating the right things and doing exercise regularly. And when I talk about exercise, I mean endurance exercise and not just pushing weights, because that's nice if you want to do that for fitness and for muscle building, but not exactly for your microbiota. For that, you have to do something on endurance.
So when I might add to that, I also have to say that the most important aspect is lifestyle. And lifestyle is a very complex process, of course, but it involves definitely the way we are eating. And I would say on a rather general way, we recommend Mediterranean lifestyle, so not too much red meat. And there are so many studies, especially with colorectal cancer and food associations, where it has been shown that red meat is a disadvantage. So modulating your diet is very, very important. Then also looking at your weight balance is very important, and exercise is very important. And probably it's a summary of all those aspects which is so relevant for our health. And it also brings back the responsibility to each person, the way you are living, the way you contribute to your health and also to your GI health. And we know, for example, from the gut microbiome, it's modulated over the whole life. So it's a very dynamic world inside, and it's dynamic until the end of life. So we can always contribute over the whole life. And there is an interesting question I've read here, which fits probably into this discussion. It has been stated here, our generation has been extensively cured when we were young by antibiotics. At the same time, we are getting older and more prone to developing colorectal cancer. And at the same time, we are the most targeted population to get the COVID vaccine. Will the three factors trigger a higher rate of colorectal cancer? And I would say yes, food is very important. The use of antibiotics in early life, we do not know exactly whether it might have a disadvantage for certain diseases. We know it has a disadvantage, for example, for the development of inflammatory bowel diseases, so a disadvantage for developing inflammation. And for the COVID vaccine, my personal opinion is it will play no role, no effect on more colorectal cancer.
It's just, if I may add to that, I think one problem in the whole pandemic and is in COVID is that people tend to postpone preventive measures. So they don't go to colonoscopy because they say it's so busy anyway, I'll wait till everything is over. And now we see it's not over in a on a short term. So my plea would be to everyone, don't postpone any preventive measures. Go to your doctor, don't go to colonoscopy, do your FIT test for preventing colorectal cancer. That's much more important than, together with all the other means, food and exercise, than being scared about having potential effects of the vaccine. And I'm absolutely with Herbert. I don't think the vaccine will not have any role in colorectal cancer carcinogenesis. And I'm not so sure about stress either, because we don't know what type of stress triggers cancer. And there might be also some stress which is good for you. So we don't know really about that, but we know about other things that are really important, and prevention is one of them.
So Thomas, may I ask here along those lines? You know, we had in one of the last webinars, we had Aaron Siegel here, who showed us the very individualized approach with regards to diabetes interventions and microbiota. So the question here on carcinogenesis would be, you know, you're giving rather general rules, but is it really one fits all, or is it more individualized things that one diet might be preventive for one person while it might harm another person? Maybe we're not there yet, but do you have any ideas of how to tackle this?
Well, generally speaking, we know there are healthy diets, and that's what Herbert already mentioned, Mediterranean diets. Also, when people change, when they go from, for example, Japan and China, go to the US and change their diets and go to a Western style diet, we see that there's a huge increase in colorectal cancer. So I think there is actually a quite general thing, what is good for you. And a lot of vegetables, fruit, fiber is good. Red meat, a lot of red meat, a lot of alcohol, and a lot of fat is not good for you. So I think this one can generalize. It might be that certain tumors are specifically prone to certain effects of particular components like polyphenols or something like that, but it's very, very tricky, as you know, to assign specific functions to a specific nutrient, because our nutrition is so various. We eat so many things, and it's over the whole lifespan, that it's very hard to really attribute certain food to certain effects. And I think this is what sometimes industry wants to tell us, that you have to eat this and that, or you have to buy this and that, then everything is fine. And that's unfortunately not the case. And this also applies to nutritional supplements and stuff like that. A good mixture, diversity in nutrients, is also quite important as compared to diversity in microbiota.
And in diabetes, obviously, it's easier because you can measure the blood sugar the same day, you know, and he could show that with 2,000 different diet modulations, you can actually have a great impact. We understand that in cancer, this is a different story. Sarah, you wanted to ask something, or Herbert, you...
Yeah, I just wanted to state also.
Erstmal Professor Tilg und dann ich.
Yeah, you mentioned the discussions you had before with diabetes and all those diseases. I mean, but it's also true for colorectal cancer. It's in a way, of course, it has something to do also with age, but it has a lot to do with lifestyle and nutrition. So it's also a nutrition-related disease. And for example, the fact that we are now seeing colorectal cancer below 50 in the whole world, that's an issue which is definitely associated with environmental factors, and we believe it's associated with diet, and it brings again now in the political dimension. And dear Sarah, it's very important because it's the lifestyle we all live and have, but it has big disadvantages. It leads to diseases, and colorectal cancer is just one disease because too much food, too much dense food, too much ultra-processed food, too much meat. It's so important. So in a way, it has, of course, a big public health dimension and cannot be dealt by physicians or by us. And therefore, it has a very important public health aspect within it.
All right, Sarah, please.
Ja, es hören ja nun auch einige politische Entscheidungsträger zu, so wie ich. Was sind denn die Herausforderungen und was würden Sie sich wünschen von der Politik, vielleicht als Rahmenbedingungen, worauf muss man speziell achten, was wäre hilfreich? Und dann würde mich aber auch interessieren, was sind denn die besonderen Herausforderungen und aber auch die Chancen oder die Hoffnungen der Mikrobiomforschung?
Herbert, you want to start and then I follow?
Maybe you can start and I bring microbiome changes then.
Okay, so the problem is science can raise awareness and society has to actually establish it. And this is what we see with the COVID vaccination. This is what we see with, of course, the microbiota as well. And I think raising awareness on healthy nutrition, also getting the prerequisites right for advertising, for example, the traffic lights on sugar content and fat content in nutrients, I think is very important to really know that everyone can immediately see what type of nutrient am I eating. It's not that you forbid people to do something, but you raise awareness and you see this is a red light sugar prone nutrient. So don't really eat too much of it. You must not eat it at all. No, that's not the point. It's not forbidding it, but it's just raising awareness. And a lot of people, if they would be more aware of what they eat, I think they would be treating themselves much better. And we have to start very early on. We have to start with the kids. We starting with older age people, it's nice, but we have to start in the schools. We have to start with school food as well, which is horrible at times, at least at some places in Germany. And so we have to really start educating our youngest to really not become an obese adult population. And this is, I think, part of the reason what Herbert already mentioned, why we're seeing this increase in younger aged people with colorectal cancer, because this is the way we treat ourselves. We treat ourselves not well enough. And I think politics has to support that, has to raise awareness, and has to really educate. Would be my plea. And research in microbiota, and this has already been mentioned by Marcus, the project where we also compare microbiota. I think we can do an awful lot. We have to also set the limitations right. This is not a novel wonder world. This is just another way of understanding how cancer originates and how cancer treatment is modulated and progression is modulated. This is not going to sort out cancer once and for all. This is one part of the puzzle contributing to the whole picture of how a tumor is actually growing and how a tumor is developing and propagating. So it's very important. It's also important to support research that these projects, as mentioned by Marcus, are for me quite important. And it's important to put that on a broader level to really compare country-wise also, not only in one particular area, but to really see what is the relevance and what is the broader level. And of course, the next level is omics, because a lot of bacteria work via their metabolites, and to really see what are these metabolites and how can they be manipulated. I think this is the next challenge, and is already taken, of course, by many groups. So there is a lot of potential. It's not going to solve the problem, but it will get us one step ahead again.
When I might continue, I totally agree. It's public awareness, and it's very similar as with diabetes or obesity. And please keep in mind, colorectal cancer is highly associated with diabetes and obesity. So those patients, those affected individuals, have a higher rate of colorectal cancer. So we will need very similar preventive strategies. Medicine cannot achieve that. We need public health awareness. We need big campaigns. Thomas just mentioned traffic lights, and I think we need a lot of actions into such directions. And maybe nicotine consume has been an easy thing, question mark. Food is much more challenging, I believe, but we need probably similar approaches so that people everywhere will understand what is maybe more or less healthy for me. And that's already a very complex question for most of patients, but it's the only chance when they go into these directions. And of course, microbiome research will be very important, because we hopefully can identify, let's say, the 10, 20 or 30 bacteria which play a key role, which contribute to colorectal carcinogenesis, and which can be manipulated in the future. I'm convinced this will be possible, but this is still a long way to go. But that's the ultimate goal of microbiome research with respect to this disease, to really understand what are the metabiomes, what are the key microbes driving this disease.
Yes, and of course, the obvious things, and I mean, this is why I highlighted the Aware Week currently ongoing, is of course, alcohol is one of the nutritions that we need to control, you know, Absolutely. And control much better. You didn't mention it specifically, but of course, there's good data showing the negative impact of alcohol on the microbiota and through that on cancer development.
I mean, with respect to this, we have nicely shown that, for example, alcohol eliminates one bacterium which has the name Akkermansia, and this is one of the most beneficial bacteria inside our body. So this bacterium is killed by alcohol. So it's a nice example how alcohol has bad effects on our health in general.
So Sarah, this would be a very important message for the politicians, because you realize that in Europe, we're really struggling to control alcohol. We don't even get alcohol labeling to a reasonable legislation. So it's really an uphill battle. And microbiome research is just one part of the whole thing, but I think controlling advertising and getting the message out is really very important from a political point of view.
Ja, jetzt wollte ich gerne noch etwas dazu sagen, ganz kurz, weil wir nähern uns dem Ende, was ich in den letzten Mikrobiom-Webinaren gelernt habe, was ich aber auch von anderen Ärzten und Forschern gesammelt habe. Mein Hauptinteresse liegt natürlich in der Ernährung. Ich habe eine Stiftung, die anfängt in Grundschulen und Kindergärten Kindern natürlich und vielfältig Kochen beizubringen. Also ich glaube, die erste Maßnahme wäre wirklich, die Körpersouveränität wieder den Menschen zurückzugeben und ihnen eine Natürlichkeit zu bewahren, denn wir haben gehört im letzten Webinar, dass auch schwerst verarbeitete, ultraprozessierte Nahrungsmittel das Mikrobiom dezimieren. Wir haben gelernt auch, dass es natürlich auch individuelle Unterschiede gibt von Mensch zu Mensch, dass wir mit einem Mikrobiom geboren sind, dass wir uns unterscheiden. Wir haben gehört, dass wir auch nicht sozusagen den Krebs und den Tumoren ausgeliefert sind, sondern dass jeder auch selber etwas Verantwortung übernehmen kann mit seinem Lebensstil, mit Bewegung, nicht rauchen, Alkohol in Maßen und besonders auch vielfältiger, frischer, natürlicher Ernährung. Ich kann es nicht oft genug sagen, weil es hier eine Nahrungsmittelindustrie gibt, die uns glauben machen möchte, dass schwerst verarbeitete Nahrungsmittel angereichert mit Zusatzstoffen die gleichen Effekt haben wie natürliche Nahrungsmittel. Das ist nicht der Fall. Insofern glaube ich, weil das auch gerade ein Thema in meiner politischen Arbeit ist, Labeling, aber auch Verbot von Kinderwerbung, die übrigens Kindernahrungsmittel süßer noch sind als für Erwachsene. Das ist ein Wahnsinn. Und es damit Werbung gemacht wird, dass man sich am Ende schon gar nicht mehr auskennt, was darf man essen, was kann man essen, was soll man essen. Ich rate Ihnen, essen Sie nichts, was Ihre Großmutter nicht als essbar erkannt hat. Essen Sie möglichst vielfältig, möglichst frisch, möglichst individuell und das, was aus regional und saisonal ist. Und wenn Sie noch ein extra Sternerl haben, dann aus ökologischen Anbau. Das war jetzt mein Schlusswort. Ich möchte gern an Markus übergeben. Ich wollte mich aber schon vorab bei unseren zwei hochkarätigen Koryphäen bedanken für diesen Einblick. Sie können dieses Webinar auch nachhören. Auch die folgenden Webinare möchte ich hier nochmal erwähnen. Und ich danke herzlichst für die Zeit und für den Input und auch für die Inspiration, die Sie mir wieder mal persönlich gegeben haben. Vielen Dank.
Sarah, thank you very much. I would also like to thank you, of course, for being the host again, and the two fantastic speakers and the topics. And of course, we could talk much longer. But I think this was the third webinar that we did. It will be my last one since I'm stepping down, but I know the initiative will continue also with with the next chair of the Public Affairs Committee. So thank you very much to all of you, and I'm looking forward to the future webinars. I'm sure they will be very interesting and very informative. And as we, the last question that we got here in the chat is how can we improve awareness, and would the European Code Against Cancer need to be updated? Well, I'm not sure that that Code Against Cancer needs to be updated, because essentially everything's there, but we need to talk more, and there needs to be a lot more emphasis on nutrition. We need to ban the advertising of harmful diets. You know, sounds like a simple step. It's very difficult on the political field, but this is, I think, for next year. And for right now, I'd like to thank you all very much, and goodbye.
Have a good time. Thank you for inviting.
Bye-bye.
Bye.